There are a lot of interesting developments in the acute leukemias lately, and there are three main areas that were touched upon at the iwAL meeting that we're lucky enough to be at right now. The three areas are leukemia biology, new therapeutics in the leukemias, and new ways to monitor the disease and assess response. So in terms of biology, we're learning a lot more about leukemia stem cells. The traditional ways of describing leukemia stem cells, it's been an ability of a subset of the bulk leukemia cells to be able to repopulate in a mouse, but now there's new technologies, new mice, and new thoughts about how stem cells develop over time. And Dr. Majetti from Stanford gave a very exciting lecture about that the other day. And so our challenge as clinicians is to be able to take that data and apply it to clinical medicine. A lot of it is the thought about whether we should sort of cover all the potential mutations that might pop up at the beginning, whether we should wait and see what actually occurs in a patient given with therapy A and wait to give therapy B until we see the development of those new mutations. In terms of therapeutics in both ALL and AML, it's amazing time to be a leukemia doctor because there have been, I think if you added up, about 14 new therapies in those two conditions in the last couple of years, or at least new indications. And so in acute lymphoblastic leukemia, we're beginning to think about application of kinase inhibitors to not only patients with Philadelphia positive ALL, in which case the ability to give BCR-ABL inhibitors has made a huge dent in the disease and maybe even obviated the need for transplant in what was considered a very bad risk population, to now where we're thinking about using kinase inhibitors in another interesting and fairly recently described subset of ALL called Philadelphia-like ALL, where the expression signature in this subtype is similar to what you do see in bona fide Philadelphia positive ALL, and whether we should be, it's this Philadelphia-like ALL, which is seen in all ages, but is usually a marker for disease resistance, whether we should approach that with specific kinase inhibitors. Some patients will respond to JAK inhibitors, others to ABL kinase inhibitors, whether we should be using that, as the pediatricians have been doing, whether we should be using so-called MRD erasers when the inevitable disease burden is still present after standard chemotherapy. In that regard, Dr. Gakibet reviewed the data using blinatumomab, the bispecific CD19, CD3 drug, as an MRD eraser, as something that can be used after chemotherapy is done, at least a few cycles of chemotherapy. Then if you detect MRD by either DNA sequencing or by flow cytometry, you come with a blinatumomab and lower the disease burden in that fashion, thereby seeming to making the outcome better. In fact, blinatumomab has been recently approved for the treatment of minimal residual disease or measurable residual disease, ALL. That's just one example. We'll be talking about CAR T cells in ALL later today, and that's been a huge new therapeutic option for some, particularly young patients with relapsed ALL.